placenta had more than 2 trophoblast inclusions in 4 different samples, whereas the high-risk autism group had up to 15 in 4 slides. So just like the dose response curve I showed you before, there is a normal range, which I consider to be 0, and then autism is just above. It has an average of 0.5 trophoblast inclusions per slide. So it seems that in fact there is a correlation. And going back to my first question, does the placenta predict the brain? Well, it does seem to. Abnormal folding in the placenta seems to be reflected in the abnormal folding that's seen in children with autism. Not only are their brains larger, but they have increased folding at a microscopic level and a macroscopic level. So MMR, surface MMR of children with autism show increased folding of these brains. What I found even more interesting about children with autism is that the airways of the lungs showed increased branching. So this is a mask, basically a mold of the bronchial tree. This is a bronchoscopy looking with a scope down a normal child's airway. Compare the normal on the left to the doublet branchings of children with autism. So the branches of the lung are actually branching more. And interestingly, people who know children with autism know that they have a number of GI symptoms. When people look at the branching and folding of the villi in the gut, there's increased folding there in the gut also, which is all background to why I'm here today. I wonder to myself, if this is a whole body situation, why is there increased folding? And could there be any advantage to that? And I realized, being an OBGYN, that one of the biggest problems that we face in terms of reproduction is this conflict, is getting the skull out of the female pelvis. And although you would have a tendency to think that having a larger head and a bigger brain would make you more intelligent, there is a very hard ceiling to that phenomenon. You cannot just have an infinitely and unfettered increase in skull size, because if you do, you won't get out of your mother's pelvis. So from an evolutionary point of view, I think what's happened is that there's been pressure to pack more brain power into this limited sphere by having increased folding. And I've shown here a diagram that, and I understand from, and I'm not an evolutionary biologist, that the ontogeny recapitulates phylogeny concept has had some, criticisms, but I think it's intriguing that when you start with a rat or a mouse and it shows no folding of the brain at all, and as you go up the phyla, here's the human right here with lots of folding, and here's the dolphin with the most folding. Now, in sympathy to Barbara's talk this morning that we heard, I just want to mention that I'll be going to SeaWorld after this conference to be presenting to dolphins because they have expressed an interest in this work also. Now, if we look at human development in the embryo, you can see there's increased folding. So what I think is happening is that this increased folding is allowing us to put more CPU power into the limited skull size we have. And just like we saw before from John, there's a bell-shaped curve for this. You can think of most normal people of having a certain brain size and a certain amount of folding, and there are 1 and 2 and 3 standard deviations above the mean, and autism is going above the mean. Their heads are larger, their brains are larger, there's increased folding. And the converse, there's an unfortunate family in Turkey, a clan, that has a 1-point mutation. Their heads are smaller, their brains are smaller, there's decrease of folding here. So I think that and I would just allow you for me to speculate a minute, I think that there might be evolutionary pressure globally to pack more brain into the skull, and that shows up in a whole host of genes which show increased folding in various organs, including the placenta as simply a marker, but its ultimate purpose is to get more brain power. So in conclusion, what we're looking at now is the basis for this increased folding, what is the biology behind it, and how can we link these things together. I'd like to acknowledge the many people who've helped me with this study, and I think I have a little time for some questions now. Thank you.
Very interesting. There was just a paper I saw published making the argument that in families that have autism, people who don't have autism have a slightly higher measured intelligence or IQ. that might go along. So the point is that the increase in intelligence might be related to genetic variants that cause autism, and in those individuals they may not have higher intellect, but they may have relatives who do.
Right. So one of the and I agree completely, and I think we all know families like this. I can even speak of my own family. They're high-energy nuclear physicists and MIT professors and people who are very successful and smart, and then floating around there are people with autism. And I think of autism as going off this curve that I showed you. When you're 4 standard deviations above the mean, you've passed the point of improvement and you can get to the point where the connections can't be made successfully. So I think I view, and I would never promote terminating pregnancies, for example, for this condition, because I think that this is where a lot of human intelligence comes from, is these genes.
Okay, we have to stop there, I'm afraid. I could see somebody perhaps making a link between this talk and why Neanderthals went extinct with their larger brains, but I just mean that as a joke. So anyway, we're moving on. We have Anne Stone now, who's on her own home territory. Anne works in paleogenetics, ancient DNA, and the evolutionary history of diseases. And she faces a trade-off today by having the advantage of being on her own home territory, but having to run off straight after this talk to take care of some of her students who are in an exam situation. So, yes. Anne, thank you. And she's going to be talking about Mycobacterium tuberculosis, origins and evolutionary history of a major pathogen.
Thank you. So I will be around in the afternoon after I finish teaching, but if you have additional questions afterwards that you aren't able to ask. So today I want to talk about some research that's going on in my lab, looking at the evolutionary history of tuberculosis. And in particular, we are interested in what the origins of tuberculosis are and really the patterning of diversity pre and post age of exploration. And with a particular focus on the Americas. And that has turned out to be a very interesting story, and we're hoping to add genomic data to the story fairly soon because we're very much interested in the question of how mycobacteria have adapted over time as they jump from one species to another or as different strains interact, and also with the improvements in genetic analyses in ancient materials, we can even go and look at the host DNA to try to look at that side of the story as well. So what is tuberculosis? It's a disease of the lungs, typically, not always, but today most cases are due to inhalation through the respiratory route. However, the gastrointestinal route is also an important route of infection. And it's a disease that's caused by a number of different mycobacterial species, ecotypes. There's debate about that. But these include Mycobacterium tuberculosis, this complex, including Mycobacterium tuberculosis, Mycobacterium africanum, M. bovis, M. caprae, M. microti, etc. It turns out that humans can and are infected by all of these, and the symptoms that are shown is the disease tuberculosis. It's treated in the same way. Most humans tend to be infected by the classical M. tuberculosis. So what are the origins of TB? For a long time, it's been thought that Mycobacterium tuberculosis evolved from Mycobacterium bovis. So we got it from cows during the process of Old World cattle domestication. We have the first clear historical evidence for TB from China about 2700 BC, and the first clear skeletal evidence from Italy and Egypt around 4000 to 4500 BC. Now, if this is indeed the case, then one would expect with the colonization of the Americas about 15 to 20,000 years ago that basically you've got people, but we don't think that cows migrated as well. And so therefore, in the Americas prior to European contact, we would not expect to see tuberculosis. However, one of my pesky collaborators, Jane Bijkstra, just kept finding cases of TB in the archaeological record, she and others. And she was also involved in a study in 1994 that used new ancient DNA methods to target the IS6110 repeat element that's found in the MTB complex. And they got a band, which was state of the art in 1994. It's like, yay, which basically says, yes, the TB complex is there. Ta-da! Which is not so satisfying if you are interested in the evolutionary history of the pathogen, you want to know more. But the methods at the time were not amenable to that. Now, parallel to this, we have increasing evidence and information from modern strains. So people in the clinical world were mostly interested in, is it something that causes TB or not? And, therefore, how do we then treat it? But people began to start looking at the genetic diversity in the strains, and what they noticed was, starting in 1998, and then with Roche et al. In 2002, is that, let's see if this shows up, yeah. Okay, so here are these so-called animal strains, and you'll notice that they're more derived than the human strains. So this kind of put a kink in the notion that cows gave us TB and actually suggests that we might have ultimately given it to them. So in the last few years, we've gotten a lot more genetic information about the diversity of M. tuberculosis strains. And this is work by Peter Small and Sebastian Gagnon and collaborators. And I want to point out a couple of things in this phylogeny, and you'll see the colors that you see in this phylogeny through the rest of the talk, representing these lineages. So the first thing to point out here is that you have this group that is possibly less virulent. It has a set of genes that seem to make it grow a little more slowly, perhaps, and these seem to be the older lineages. You'll also notice that the animal strains here in orange are nested within the West African, two different West African lineages. The greatest diversity of strains is in Africa, so we think the origin is there. But today, when we look at the animal strains, they're all nested within the human strains. So we don't actually know what the origin of tuberculosis is. Now, we also see this group here, which has this deletion that appears to make them more virulent. And these are the strains, including the Jing strain and others, that are spreading today. And so Gagnon et al. And others have suggested that those strains may have really taken off with the Neolithic transition and increase in population size. So our questions have been, what was going on in the Americas? Is it the case that perhaps this old Asian strain that's pink maybe came over with people? By the way, the timing on this has ranged from very recent with Neolithic all the way back to 3 million years for the origins of tuberculosis, or the time when it jumped into humans. So there's a huge span, or there was until recently a huge span there. Or it was the lesions that we see in the Americas pre-contact caused by something else, in Kansas and something else. And then, with the age of exploration, you have basically the movement of these more virulent strains around the world. And so today in the Americas, what you see is the European strains. In big cities, of course, you see worldwide constellations of strains that reflect the people who have immigrated. So what we've done is sampled skeletal remains with evidence of tuberculosis. These have very characteristic lesions. Keep in mind that the vast majority of people who develop tuberculosis never develop skeletal lesions. So these are people who have survived with the disease chronically for a long time. And then we grind up bone and extract DNA. We have a clean room here at ASU that's a class 10,000 clean room. My grad students have to constantly shower and wear this garb, so they're a very squeaky clean bunch. And we work under these conditions to really reduce the possibility of contamination. So our strategy initially is to extract DNA and use quantitative PCR to test for three different sets of markers. The first is IS6110. Actually, the first here is IS1081, which is found in 5 copies in all members of the MTB complex. IS6110, which is found in 0 to 27 copies, and then RPOB, which is a single copy gene that's found in many bacteria, but we can use very specific probes and primers to target it. So when we do this, we first did this using a probe that we call RPOB1, and we realized, we were very excited because we had 31 that tested positive, like yay, excuse me. But then we sort of started to notice that this particular marker was perhaps much more positive than it should be, given that it's a single gene marker. And one of the real challenges with ancient DNA is that when you're extracting DNA from a bone, you're extracting all the DNA from the bone. And mycobacteria are also found in soil and water, non-pathogenic forms. So we have to worry about whether or not our assays pick those things up. And so we had designed this in about 2005. We went back to GenBank to get more data from all the accumulating strains. And we found that new species had been described, some of which might be giving a reaction for that. So we redesigned the probe, which is now called RPOB2, and you can see that it's much more in line with the other evidence. So then, if you disregard RPOB1, we have 12 samples that were positive. So the next part of our strategy is to use capture to pull out the DNA of interest, so specifically the MTB genome. We first use a capture enrichment that is a in-solution hybridization for 5 genes and sequence those usually on the MySeq. The second thing that we do in samples that work well for the 5-gene capture is we use an array enrichment to target the entire non-repetitive genome. We've made sort of this ancestral genome that puts all those deletions back in, and we've tiled that on 2 arrays. It takes up 1.5. The rest of the array, we put some M. kansasi and M. avium, which also can cause variants of tuberculosis. So we reported last fall our first genome sequences from 3 samples from Peru. Since then, I'd hoped to be able to give you a hint about these. We just got these additional samples, came off the HiSeq about a week or 2 ago, but they're still in the pipeline. So I email Johannes Krause, who I'm collaborating with every day, and I'm like, any word? What's the results? But I can tell you what we found previously. So all 3 of these samples happen to be from the same valley in Peru, from 3 different archaeological sites that date to about 800 to 1,000 years ago. It's a very dry region. The preservation is amazing. This is one of the burials. And what you can see is that when you look at these 5-gene capture, you get, this is just for the, no, it's all 5. You get pretty good coverage, so this is why we put those on the array. And we also see the very characteristic DNA damage patterns, which is sort of U-shaped. And this is reflecting the overhangs that tend to be deaminated. So here were our results. And because we had radiocarbon dated samples, we were able to calibrate the timing of the tree. And what we were very surprised to find is that the Peruvian tuberculosis genomes cluster with M. pinnipedi. So these are TB strains found in southern hemisphere pinnipeds, seals and sea lions. So that was quite unexpected. So what we propose is sort of the overall view here is we have an origin of TB in Africa, somewhere in the neighborhood of 3,000 to 6,000 years ago. So much more recent than we had originally thought. We have no idea what the source is. We then have the spread of these more basal lineages, a jump back into animals at some point and somewhere. Then a new lineage, which seems to be basal to the more virulent ones, arises. The more virulent lineages arise. You have the jump from an animal into pinnipeds. They swim across the ocean about 1,000 to 1,300 years ago. Give it back to us. thank you very much, and perhaps have spread it through the Americas. So currently we're really targeting, were there multiple jumps? Did the pathogen sort of show evidence of readaptation to humans? And we do see 5 non-synonymous SNPs that differentiate the human strains from the impinipedi strains. Did it spread via human-to-human contact into North America? And so we are expecting new samples that are actually from North America, so I'm very excited. And the other possibility, of course, is did TB reach the Americas before 1492 through other means? And I'm very excited because I'm going to be getting some cheek swabs from Antarctic seals shortly. But then we think after Columbus, these more virulent strains were the ones that were placed. So this is the final slide showing you kind of what we think happened in terms of the evolutionary process. So with a replacement event, and with that, I will thank my many collaborators.
Okay, thank you, Anne. I'm afraid there's no time for questions at all, so we have to move straight on to our next speaker, who is our second European representative, Martin Hausler from the University of Zurich. The University of Zurich was in fact the first institution that set up a Center for Evolutionary Medicine with a focus on functional morphology, anatomy, and you'll see on your tables there's a little notice advertising a sister meeting that they're going to be running in July of this year. Okay, and Martin's going to be talking about our musculoskeletal.
We find almost no hip osteoarthritis in them. Also in human prehistory, hip osteoarthritis is extremely rare. There is only one case in a Neanderthal and a second case here in an Australopithecus africanus from Makapansgat, South Africa, a 2.6 million year old femur fragment. Although it is quite fragmentary, we nevertheless clearly see the characteristic signs of osteoarthritis, including these large osteophytes. Because the head is split apart, we also can look inside the head and see these large cysts, and Here there is a osteophytic cap, which is medially located rather than superiorly as usually, and this is best compatible with protrusia acetabuli.